Welcome to the Expert Insights webinar titled Wisdom from the Front Lines of Successful NDA Submission Programming, Utilizing the Biometrics Operational Data Model. We appreciate each of you joining today. My name is Don McLean, Senior Manager of Global Marketing and Communications at MMS, and I'll be your moderator for today. Now, I would like to introduce our three panelists. First, we welcome Jim Badamian, Global Head of Statistical Programming at MMS, based in North Carolina. Jim has more than 20 years of experience in statistics and programming and was formerly Associate Director of Statistical Analysis and Reporting at Pfizer, among other positions. Welcome, Jim.
Thanks, sir.
Next, we have Sushil Arkala. Sushil is Principal Clinical Programmer and Associate Manager of Programming at MMS. He is based in Portland, Oregon, and has been working on regulatory submissions within the group for more than 8 years. Thank you for joining us today, Sushil.
Thanks, Don. Excited to be here and looking forward for the discussion.
Wonderful. Lastly, we have Chris Hurley, Senior Manager of Global Data Operations, Standards and Systems at MMS, bringing more than 20 years of programming, data management, and stats experience in the pharmaceutical industry. Prior to joining MMS, Chris worked as Associate Director of Statistical Analysis and Reporting at Pfizer Global Research and Development, as well as Manager of Clinical Data Programming at the former i3 StatProbe. Currently, he serves as the U.S. Events Director for FUSE, the Pharmaceutical Users Software Exchange Organization. Thank you, Chris, and again to all the panelists for your time today.
Thanks, Don. It's a pleasure to be here.
All right, without further ado, let's begin. Chris, I'd love for you to start with the Biometrics Operational Data Model and explain how submission work can be streamlined across programmers and statisticians.
Okay, thanks, Don. I'd like to start with this graphic here. We've got a swim lane diagram. We've got 4 lanes, one for the analysis team, one for the stats team, the study data team, and the eSub team. Now, applying this model enables you to get all 4 of these groups working together and working efficiently right from the get-go from your project. If it's a submission project and it is a single study, then we could start with the study data team. So they develop annotated case report forms and specifications. They create the data sets using macros. They create the data sets that are needed for a submission if we're going to do any integration. So we would focus on the data sets for the safety and the data sets for the efficacy if those were needed. Then once those are set, the analysis team can take those data sets and create the statistical analysis plan for the individual study, or the ISAP integrated for the integrated summary of safety and integrated summary of efficacies. So using the ISAP, they can develop the shells, or if it's just a SAP, they can develop the shells for the study. And so what happens there, once we've got the shells, we can go ahead and develop the ATOM dataset specifications. And then from those specifications, we have some more macros and programs that will create the data sets. The data sets will be reviewed by a statistician before we go ahead and develop the TFLs and create the final TFLs, be it for an ISE, an ISS, or for a study. Now, as this goes along, we could have an eSub team who is working on legacy data. So if we were talking about creating all of the legacy studies, if they were going to submit those, we'd like to know, we'd like to get started with those immediately from the get-go. So what we could do is we could get a team working with the legacy data and the data sets. They could upgrade any of those data sets to comply to field lengths, field names, create proper SAS XPT files, which we call legacy tabulation data sets. We'd also create the case report tabs and the defined PDF. So we could put that information together in parallel while the other groups are working on the standardized study data with using the SDTM and ATOM models. So once we've got the legacy data, the SDTM data, and the ATOM data together, we can create the reviewers guides. So if the SDTM data comes together first, we'll take a look at those data sets from a programming study data team, and we'll pass the reviewers guides and some of the information that we've gotten from these data sets to the stats team, who will then review these data sets for submission, just to make sure that they're good, they're compliant, and good enough for the analyses. Then once those are set, the reviewers guides can be created and then appended within the define XML. The final define XML can be ran and then create the define for all the tabulation data for this SDTM data. What we do when we create those define files is we run them through the Pinnacle validation to ensure compliance, so that when they're loaded into the FDA site, there are no problems with these files. If we're talking about ATOM data, now we would review the ATOM data through the stats team, and they would do the same thing. If there are anythings to add to the reviewers guides, they would add those. They would make sure that the data sets comply, and then those data sets would then be ready for creation of define XML and for the Pinnacle 21 community validation. So once those data sets are done, we create the define XML and we append the ATOM study data reviewers guide into the define XML application for ATOM. When all that is done, when the data sets are done, we can have the stats team and the analysis team develop the tables, figures, and listings. And once the tables and figures and listings are developed, again, those get reviewed by the statisticians, and final TFLs are created. We have a QC process that we apply on top of the validation. We do parallel programming validation, but every item gets QC'd before it goes to a sponsor or before it gets included into a submission. So in this case, you can see here in these white swim lanes, that can be for a single study, or it could be for integrated summaries, and where things can be developed in parallel. And that's pretty much it. So we have everybody working together effectively, efficiently, and from the get-go using this model.
Thanks, Chris. Appreciate your time speaking to that. Jim, what exactly needs to be validated within a submission, and how should it be done? And as a follow-up, how does third-party software like Pinnacle 21 come into play?
Thanks, Don. I'd love to tell you that the validation's easy, but there's really no shortcuts here. Everything needs to be validated. Parallel programming as normal, independent, meeting specs, not just meeting and matching. We want a two persons independently program and match at the end, obviously. The third-party stuff is great because it provides some consistency checking and adherence to the current standards, and really will find things that parallel programming might not find. So it's always a nice add-on. Chris or Shashil, anything to add on that one?
Yeah, I mean, thanks, Kim. Just to add to that. Apart from just parallel programming, and once you have programmed your data sets, or once you have programmed your TLFs and ran through your third-party validator checks, there are some other validation checks we can perform, like in a submission or an in-hand study. We can always cross-check some of the numbers within the study or within a submission. Or if you are going for a submission, then you can always cross-check your ISS and your ISC numbers with your individual study CSRs. Technically, in a perfect world, your individual study CSR table numbers should add up to your pooled table numbers. In case if those are not adding up, then it's good to know why it isn't, and then just have a documentation ready, just to be able to explain to the reviewer, or just putting this in reviewer's guide.
That's a great point.
There's another thing to consider here is the data provenance, or the origin of data, right? So we like to use the Pinnacle software because it has all the rules that are applied based on the data models that are used for the data and for the XML application. For instance, we'll run Pinnacle 21 for Define XML, and it'll give us any messages if we're missing something. Say we're missing those reviewers guides, we'll get a message. If we're missing controlled terminology or value level metadata, it'll show us, what's missing. So we can validate. Sometimes things get through, but with this third-party validation, it really is a good check and very useful.
Thank you all. Moving on to our next question, Sushil, maybe you can speak to reviewers' guides and why they should be built well before the NDA is even planned.
Oh, thanks, Don. And I mean, as we all know that in a submission, we submit your data sets, your programs, and your define.xml in your ECTD module 5 folder. And sometimes all these documents or your data sets doesn't adequately support your SDTM mapping decisions or any of the analysis that has been done in that particular study. Or why a particular subject has been treated or a group of subjects has been treated in a particular way. And that is where this reviewer's guide kick in. And FDA collaborated with some of the working groups like FUSE and CDIS. In collaboration with CDIS and FUSE, they have come up with some templates which are available on the web. And we have got like two different reviewers guide. One is study data reviewers guide, and another one is analysis data reviewers guide. And in the study data reviewers guide, normally you put in the SDTM, anything related to SDTM. Like just a quick summary of it is you put it your protocol design, a short summary of your protocol design, and then if as Jim was mentioning earlier, like you would run your Pinnacle, a third-party software like Pinnacle 21 on your SDTM datasets, and sometimes the report doesn't come out clean, and there would be some errors in that, and there is a valid reason why those errors are popping up in your Pinnacle 21 report, and the reviewer's guide is a perfect place to justify your reasons, your conformance issues, and one other example is that you have your trial inclusions, and sometimes your inclusion-exclusion criteria exceeds more than 200 characters, and which shouldn't be the case. And when it exceeds 200 characters, we try to summarize them, or we try to shorten them into a meaningful sentence. But the whole elaborated thing, which is exactly as in protocol, could go into reviewer's guide as an appendix. And this is and any other data issues, sometimes like after the data was logged, you realize that there was a date which wasn't fixed in your database, and yeah, that could definitely be documented in the study data reviewers guide too. And when it's coming to analysis data reviewers guide, there are some analysis considerations that you do on a study by study basis or a case by case basis. Like there could be some sites which needs to be neglected just because of the questionable data in that, and that could always go. I mean, apart from CSR, then this could always go in your ADRGs too, and mostly about your submission programs. Like you submit your programs, and the reviewers might want to rerun your analyses, and sometimes they actually doesn't know which program used what macros, and so this is a perfect place where you have a tabular column explaining this data set is being generated from this program along with these macros. And in addition to these things, like recently, FDA has mandated a legacy data conversion plan. And as we all know, when we work with your legacy studies are normally just done. CSRs are written their legacy standards or sponsor standards, and just when they are pulling, when the sponsor is going for a pulling, they realize they need SDTM, and then after the fact, the CSR was written, they they just try to create SDTM data sets or ADAM data sets just for the sake of pooling, and in the process of creating those data sets, sometimes you encounter some issues which doesn't match the numbers when you are generating numbers from these SDTM data sets compared to CSR numbers. In these cases, then this legacy data conversion plan helps you to document all the differences from the CSR, and when you converted this SDTM, and if you had to make any corrections when you have converted this data into series standards. So like this could go along with your SDRG or ADRG as you see fit, like whatever you are creating. If you're creating SDTM and ADAM from the legacy data, then you can add it to both your SDRGs and ADRGs. And this is one reason that like reviewers guides are really important, and this needs to be set up very early in the process when the studies are ongoing. It's a good practice just to start this reviewers guide when studies are ongoing and just document everything before we forget. And it's just good to have this, and this provides us a very good guide for the reviewers, and this is a perfect opportunity for the programmer to explain what has been really done with this data. Chris and Jim, anything else to add?
Yeah, I'd like to just say that, it's good practice to keep a validation log when you're creating your data sets, whether they're legacy or SDTM or ADAM, it doesn't matter, but because if you try to keep your anomalies through email and stuff, you'll lose things. So, but even if you do keep a good log, later on if you wait to do your reviewer's guide, the data just isn't. I mean, it takes you a long time to get back into get into the swing of the data and to really understand the data. So, to do the reviewer's guides as you're going along it's just smart because it'll save you time in the long run.
Wonderful. All right, Chris, maybe you can take this one. Are there any reasons for considering the NDA submission when you're only in early phase studies?
Yeah, and just thinking about some of the things that Sushil was talking about, when you think about doing the data, creating the data for your submission, it may not be necessary, but when you project yourself years into the future, and if you decide that you need this data, now you have to go back. Now, he mentioned creating data sets for an integrated analysis. Imagine having to go back and make some data sets because you needed to include it with the submission. So you really want to check the data standards catalog at the time you're developing your data sets for your study, and check what standards will be supported. When you think if it's possible to have a submission, you might not know if it's ready yet, or if the data plays out that it's worth going for a submission. But if you develop with that in mind, you'll apply the correct controlled terminology, the correct data models, the correct data exchange standards like the defined XMLs. You'll have stuff in place so that when the time comes, should you go to submission, you'll have everything that you need. And it's not that much different than just creating with whatever standard you thought you were going to do. Well, just think ahead. A little planning goes a long way, and it'll save you time in the long run.
Thank you, Chris. All right, moving on. Jim, what can you say about trial domains? Why are they needed? How do they relate to SDTM, ADAM, or TLFs?
Well, thanks, Don. This is a great follow-up to the previous couple questions when you're, considering data. The trial domains are very simple: TA, TE, TI, TF, TV. We all know those. Some of them can be skipped when you're doing data sets and TLFs. Some are incorporated in trial visits, of course. You can use TI to get that information into SDTM data sets. You can also code around it. What we want to do is put these together now because they contain little details from the protocol or from the SAP, and it's much easier to do when your hands are on the trial and you've got all these documents in hand. If you wait, it's really a little bit longer when you've got 20 studies and you need to create these and you're looking at trying to find all these old protocols. So I really encourage you to do these now or as you're doing the studies themselves.
Just another thing to note there is as you're doing the studies, you're usually working with a team, and you've got somebody with you, maybe one of the clinicians or somebody on the team that really understands the science and the drugs involved. And things like controlled terminology, as programmers, we'll look at these Unicodes and there'll be many, and which ones do we use? Well, when you have the team assembled, it can be just a single question in your work group meeting. Hey, which code should I use? Bam, done. And you put that in your TS domain, and you're all set. So it really saves time. Just do things as you're doing them. Get these things done, and you'll save time in the long run.
Right. Well, and most importantly, go ahead, Sushil.
Oh, yeah. I was just adding to your point. Information in TS and in TVs, like this could be used in SDTM programming or in ADAM programming. For instance, like you can always use trial visits domains to map visits in your findings domains or visit-based domains. And sometimes you need the if your pool contains different indications data for the same compound, then you can get that information from your trial summary data sets directly, so that you don't have to manually do some if conditional statements. If this is the study ID, this is the indication. So just using the information that already exists in the trial domains and putting that information in your analysis even sometimes helps.
Right. And one thing I forgot to mention, by the way, it's a rejection criteria. So you have to have a TS domain, and not only for current studies using full standards, but for all legacy studies has to have a TS domain, or your submission will just get rejected. So might want to consider that.
Oh, thank you all. Sushil, maybe you can jump on this one for us. What's harder to put together for a submission, legacy data or pooled ADAM data sets?
Well, I mean, there isn't a binary answer here, but I would say that both of them has got its own sort of difficulties, and mostly when you deal with legacy studies, there is a lot of unknown information. It's being done in the past, and sometimes when people doesn't do proper documentation, then you don't know why certain things have been done in a certain way, and figuring it out would take a lot of time. And again, once if you're doing in series submission, as mentioned earlier, then what happens is you'll have to convert your legacy data sets into your series data sets, either SDTM or ADAM, and when you're converting them, then you'll come across any of those issues. If you come across any issues, then those need to be documented, and we need to either have a note to file, or either just document in reviewers' guides. And when you're pulling all this data, then you see some inconsistencies across, and then and that makes a little bit tough the legacy data. But when it comes to pooled ADAM data sets, even though let's say if you have two different studies coming from two different organizations for this for the same compound, and what happens is since SDTM has got a defined set of rules, whereas ADAM doesn't have a very defined set of rules, and you got more liberty there, and you can definitely it's a preference thing, and people can just set up their databases as they see fit now. And what happens is if data is coming from two different sources, and even though it's series standard, when you try to pool together, the variable names differs, and even though it's the same information, that the variable name differs, that either makes us to program around it, or again it makes us think that, okay, is it worth spending time in just updating all these things, or should we just go back to SDTM and program from it? So both of them has got its own difficulties. Chris and Jim, do you want to add anything there?
Well, yeah, Sushil, I just want to take a little different bend on this. I'm a strong proponent of cross-functional communication. Don't work in silos. Talk to your teammates. Like, grab a medical writer, tell that medical writer, this is what's going on. I've got these analyses. They're very complicated. If you have a small number of studies, maybe the medical writer can just do a side-by-side comparison. You might not even have to do an ADAM data set. So that would save you a ton of work. So just involve your team. Let everybody know you should all be working together anyway, and see see what they think.
That's a really good point, Chris. Maybe you can take this question then. What standards have been used in submissions in your experience? And maybe you can tell our audience some things like the number of studies involved, legacy and integrated studies, and the like.
Sure, I can give you some examples. Now, I've worked for very big pharma, and I've been in the industry a long time, well before CDISC was implemented. And I can tell you, big pharma has a lot of inertia when they're working on their own data standards, and they don't want to change, okay? And so they'll use those standards to the NDAs if they could, right? So what I'm trying to say is there's a lot invested in the standards, maybe even in the reporting for those standards, and so lots of resistance for change, okay? But the FDA has mandated, according to the data standards catalog, the submission standards, the data set standards, everything that we need to know and submit. So companies are changing. And in the last 8 years, to give you just an example of the number of studies in some of these submissions, we've had some very large submissions, 70 studies, multiple indications for like psoriasis, rheumatoid arthritis, and others. We had another submission that's 56 studies with multiple indications: pain, epilepsy, generalized anxiety disorder, just a ton of work. And integrating studies like that for a submission is an incredible effort, and it can't be done without good standards. And by implementing the CDISC, we're fully supportive of the CDISC standards. It is good not only for the structure, but for the values of those data, the data within those data sets. So I'm talking about like controlled terminology and things, the things that allow you to analyze the data across studies. So just in short, before December 17th, 2016, if your study started before that date, you could use your legacy standards. But everything after that date is required according to the data standards catalog. And there are other standards too. Therapy area user guides, so implementation guides. Those will help you if you have a certain if you're working with certain types of data. You can go to look at those standards, and they will show how data should be collected and then databased within the SDTM. So going to the CDISC site, cdisc.org, looking at the foundational standards, looking at the therapy area standards, it's a great place to start, and a great place to find the information you're going to need in order to have a good submission.
Wonderful. Did anybody else have anything to add on that one?
Well, there's also product-specific standards out there for certain drugs. If you're going to do a generic equivalent with bioequivalency. So look and do some research before you start jumping in. Make sure you're prepared.
Thanks, Jim. Well, moving on to the next question, maybe you can help us with this. What are our top considerations that sponsors often overlook as they start planning a submission?
Well, we talked about this a little earlier with Shashil. I think had the question of what's harder, the integrated atom or the legacy. For me, it's all about the legacy. Don't underestimate the time it takes to get that legacy data straightened out, in order, up to standards, mapped. It takes a lot of time. It's very tedious. You're going to do a lot of research. You know, you could have to create databases from some of these studies. They may be so old. Another consideration is try to think about what's going to happen after you submit. Maybe anticipate some review questions and do some work in anticipation of that. Maybe putting together a few extra ATOM data sets while you're in the process or shortly after submitting. Maybe a concomitant medication data set, you know, put together ABCM in anticipation of answering some of those type questions. It could save you a lot of time, and you want to do these things, especially if you think the data set is going to take a lot of time to assemble, and you get a request for information, you might have a week, and you don't want to delay your submission. So put together some good information and data sets in anticipation of questions. Guys, anything else?
Yeah, I was working on one of these compounds, and there were a lot of questions, health authority questions about hemodynamics, and we had all the information within the data sets, the analysis data sets, and because that information was there, we were able to create a lot of graphs. It takes a little while to create graphs, you know, and make things right. But we didn't have to worry about the data. The data was there. So it gave us the time it took to answer the questions. So have that data ready and think forward. When you guys are creating your data sets, your analysis data sets, think what other questions might be asked, you know, when you're developing your specifications. And if you have the data, you know, get it in there. Put it in. It can't hurt to have more than the bare minimum, because it might save you time. Again, I'm all about saving time.
Yeah, and just as I was mentioning earlier, like if you're getting data from different CROs or different organizations, then consider consistency always. Like you can always, the sponsor can always create a standard ATOM template, and so that it is used across all the studies, so that they don't have to program around those ATOM data sets or those SPTM data sets when they're pooling them together. So it's consistency is the key. Like as much consistent as we are, then it definitely helps and reduces a lot of effort when you're pooling the data together.
That's interesting. So Sushil, where do you go as a programmer or statistician when you have questions about submissions?
Don't you think nowadays Google is answering everything? With Google Home, with Siri, all they're on, I think it answers all the questions. Jokes apart, but there are a lot of things on the web. Like nowadays, FDA did put a lot of information on the web out there. They have data standards catalogs if you have any questions regarding data standards. And there is, from quite a few years, they have come up with technical conformance guides, which has got domain-specific information, like how to submit the data, like how should you submit your software programs. Those all information is in the technical conformance guides, and there are working groups out there, and another thing is one should always make use of their end of phase 2 meetings and pre-NDA meetings, and I know those are mostly for the clinical decisions or any of those very much clinical decisions. They use those meetings more for clinical decisions or more of when moving from phase 2 to phase 3. But it doesn't hurt if time permits. Like they should definitely consider asking questions in those meetings too. Jim and Chris, like, anything to add on that?
Yeah, speaking of meetings, watch an AdCom meeting. Those are very interesting. You know, it's always fun to watch and to see how those flow. But I'm a big proponent of networking. So I'm America's director for Fuse, so I'm all about networking. Go to conferences, join working groups, meet with people, get a network together so when you do have questions, you can find your answers. You can go to Pharmaceutical, a lot of papers. You can, Sushil, like he says, Google, find the answers there, but nothing like talking with a real person. And if you have a good, strong network, then yeah, that's where I start first. I usually go to my network of people.
That's really important, Chris. It really is. I'd like to end with one final question for the panel. Any last words of advice for those who are just beginning submission programming?
So,
Sushil,
yeah,
I
I
think
can
we'll start
start
with you
with
if you'd
that.
like.
Yeah, go ahead. Yeah. Like, one thing I can always say is, like, start your pooling up front. Like, you don't have to wait until you are going to an NDA. Like, once you're done with your phase 1 studies and once you're getting into the phase 2 studies, then that's when you can set up your databases, like start pooling it. When you start pooling upfront, there are 2 advantages to it. One thing is you get to see the consistency across the studies, and another thing is you will definitely considerably reduce the amount of effort that takes after the fact your last phase 3 study is done, and you have to pull the data. Like, rather than just investing time there, like, you can invest time up front. As studies are completing, you can just add them to the pool, and then once your last phase 3 study is done, then just you use the pool database and generate your tables, listings, figures as the seem fit, and we should be good for submission. And just speaking a pure programmer's mind, like, set up project level macros. Like, a simple example would be just to create a macro for a visit windowing, like where let's say your visit windowing applying to your ECGs, your vitals, and labs, and if you're doing double programming, which we definitely advise, as Jim was mentioning earlier, like, we have to definitely double program. Then you're looking at 6 different programs, and if the same logic is being used across 3 studies, then you're looking at 18 different programs. Rather than that, just set up your project level macros upfront, and then just have one on the development side, another one on the QC side, and so that you reduce a lot of redundancy of these things. And even if there was a decision made to change anything, and if it affects multiple studies, then you're making changes in only 2 different places, and boom, you run them, and like everything is updated. And that's one thing to definitely consider. And another thing is documentation. I know it's really pain to just sit and write a lot of documents. I mean, we are programmers, we just want to sit and code, but documentation is very important, and you need to document all the issues you see in a study while and when you really see it, not leaving them until the end. It's really important, even if you think it might be a minor thing which you understand, but the reviewer might not easily able to understand that issue. So it's always good to definitely document anything. And the more you spend on documentation, you might spend more time here in documentation, but that would lead to less time for reviewing, and eventually less time to get an approval from FDA. Yeah, Jim and Chris?
Yeah, those are good points, Sushil. The one thing I want to emphasize is when you're putting together your ISAPs and your analysis plans and your TLFs and designing your analysis, make sure included any industry consultants, therapeutic area experts, get those people involved early so that they get their inputs heard, and we can build those into the specifications right from beginning and get those into the ADAM data sets. This will save you a lot of time in the long run. Chris?
Yeah, here's a little homework for all you programmers out there. Go check out the ECTD triangle. Okay, that shows you what are the components of a submission. And then you'll begin to understand how these things are sent in. And I would say find a Reg Ops person in your organization, or go talk to a Reg Ops person in your network. and just ask them what it takes to put a submission together, because when they're involved, it's usually at the end, at crunch time, and literally thousands and thousands of files go into this submission. So work with your reg ops and just see what's needed, and don't wait until the end. Talk to somebody today and find out, and then you'll see what you're working towards, in the day-to-day, and it'll really hit home the contributions you make as a programmer.
Thank you, Chris. Wonderful. So I'd like to thank Chris Hurley, Jim Badamian, and Sushil for joining us today. If you have any questions about this webinar or any of the content, please feel free to send us an email at MMS or at media@mmsholdings.com, and we can connect you with one of these experts. So thank you for your time today, and please be sure to like MMS on LinkedIn for more frequent updates. Have a great day, everyone. Bye-bye.